Hello everyone, welcome to Get My Class. Let's solve this question which says arrange the following steps in correct order that are involved in recombinant DNA technology. The first statement says isolating genetic material, second statement PCR for amplification, third statement restriction digestion, enzyme digestion, and fourth statement says inserting recombinant DNA into the host, and fifth statement ligation, and the sixth statement says isolation of recombinant cells. Now they have given the code where they have given option A, B, C, D, and they have given various combination of the statements, whatever, or steps, whatever have been given. Okay, have been given. Now let us understand the question. It says arrange the following steps in correct order. So we have to arrange these steps in correct order that are involved in what? rDNA technology. What do you mean by rDNA technology? rDNA technology is nothing but recombinant DNA technology. Recombinant DNA technology. Okay, DNA technology. That is what is rDNA technology means. Now, recombinant DNA technology, which involves the synthesis of or creation of a recombinant DNA. That means the DNA where the gene of interest or a foreign gene has been inserted. That is what is recombinant DNA. Okay, and this recombinant DNA will be inserted in the host cell, and further this has to be expressed. Okay, this will be expressed here in the host cell. And what do we get when it gets expressed? Of course, we do get the products, because that is the reason why we are expressing it, right? Yes. Now, to have this, to in this particular technology, that is recombinant DNA technology, the first step what is necessary is to isolate the genetic material itself. That is isolation of genetic material. Genetic material. Okay. So genetic material, here we are talking about the entire DNA. Okay. And further, what we have to do, we have to treat this genetic material with restriction enzyme. Okay. Restriction enzyme. Okay, there are two things what is involved here. One, restriction enzyme is necessary to isolate the gene of interest. Okay, it will clean at specific site, which are called as restriction site, and you will get your gene of interest. Okay, gene of interest. Now, this gene of interest will be inserted into a carrier, which is called as a vector. And further, this vector will be inserted into the host for the expression. Okay. Now, this gene of interest can be amplified. If you want to amplify it, that also can be done. Okay, that is amplification by something called as PCR. Amplification can be done so as to get many copies of this gene of interest. Okay, and that is done by PCR. Okay, and then further, what we have to do, that has to be inserted into the vector, and this vector also can be amplified. Okay, and this will be inserted in the host cell. The recombinant vector will be inserted in the host cell, and that will be expressed. Okay, this will be expressed. Okay. Now, let us see. The first statement says isolating genetic material. Yes, that should be the first step. Second says PCR for amplification. So PCR for amplification is needed only once you isolate your gene of interest, and that can be done by the restriction enzyme digestion. So the second step should be restriction enzyme digestion. So first step should be isolating the genetic material, and this should be the second step. Okay, and then the third step will be amplification of the gene. Okay, that would be PCR for amplification. This will be the third step. Okay, and once amplification is done, what do? We insert the gene of interest in the vector, and that is done by, again restriction digestion will happen, and that insertion is called as ligation. Ligation is what? It is connecting or joining the foreign DNA with the vector DNA. Okay, so this is joining or the gene of interest with the vector DNA. That is what is ligation. Okay, so you can say this is the fourth one. Okay, then after this is done, what do? Inserting the recombinant DNA into the host cell, because Now what you have done, you have isolated the genetic material, you have isolated the gene of interest, you have already inserted into the vector, suppose this is a vector, in the vector you have ligated it, and now this vector has to be inserted in the host, and that will be your fifth step. So this will be your fifth step, that is inserting recombinant DNA in the host. Okay. And then further, after you insert in the host, you have to look for the recombinant cells, the cells in which these you can say vectors have been incorporated, or which will carry our foreign gene or gene of interest. So those cells are called as recombinant cells. So this is the sixth step. Okay. So let us see. So according to our explanation, it should be first one should be the first one, and the second one should be the third one. Okay, that is 1 3 1, then 3, then it should be 2, and then it should be 4. Sorry, 1 2 3, then it will be 5 here. Okay, and then it should be 4, and last it will be 6. Okay, that is 1 3 2 5 4 6. So let us see where it is. So A says 6, so in the beginning itself it is 6, so we can rule it out. B says 1 2, so here the second step should be the third one what is mentioned here. Okay, hence it is ruled out. Option C says 1 3 2 5 4 6, so that is correct, which is matching with our answer. So that should be the correct answer. Option D says 6 again, which is the last step, and they are showing in the beginning. So it is ruled out. So what is the correct answer? The correct answer is option C. I hope you understood the explanation. Thank you, everyone.